Good afternoon, everyone. Thank you for attending today's webinar. My name is Joel Hewitt, and I'm a subject matter expert here at the Homeland Defense and Security Information Analysis Center. We are very pleased to present today's webinar on predicting antibiotic resistance, moving from reaction to preemption of emerging pathogen. Our presenter today is Dr. Yousif Shamoo of Rice University, where he serves as Professor of Biosciences and as Vice Provost for Research. We will introduce him further in just a few minutes. The chat function is enabled in the top right-hand corner of your screen. Please feel free to type in any questions as you go along, and our presenter will address them during a question and answer session at the end. Also note, we are recording this webinar, and the video webcast will be made available online tomorrow. A copy of the slides will be available for download at the end of the presentation today. HDIAC is a Department of Defense-sponsored entity, one of 3 information analysis centers. Organizationally, we fall under the Defense Technical Information Center, or DTIC, and the Assistant Secretary of Defense for Research and Engineering. Our mission is to be the go-to R&D, S&T, and RDT&E leader within the Homeland Defense and Security community. We achieve this by providing timely and relevant information, superior technical solutions, and high-quality products to the DoD, HDS, COIs, and COPs. In doing so, we are able to help solve the most challenging technical problems facing the government. We pursue this mission across 8 discrete focus areas: alternative energy, biometrics, CBRN defense, critical infrastructure protection, cultural studies, homeland defense and security, medical, and weapons of mass destruction. Our external subject matter expert network is a critical tool in achieving this mission. And if you have expertise in one of our focus areas, we very much encourage you to apply. Our SMEs help us provide the military and government with the most up-to-date and cutting-edge information. Today's topic is of the utmost importance for national security and even global security. In April of this year, the United Nations reported that drug-resistant diseases already cause around 700,000 deaths annually worldwide. Without a sustained effort to contain antimicrobial resistance, they warn, approximately 2.4 million people in high-income countries like the United States could die from such infections by 2050. So we are especially lucky to have Dr. Shamoo here with us today. He serves as the Vice Provost for Research at Rice University, overseeing strategic planning for the university research enterprise, including engagement with industry through research and technology transfer since 2014. He was appointed Professor of Biosciences in 2012 and first joined the Rice University faculty in 1998. Dr. Shamoo served as Rice's Director of the Institute of Biosciences and Bioengineering from 2008 to 2014, leading the coordination of interdisciplinary research among Rice faculty. He received his PhD in molecular biophysics and biochemistry from Yale in 1988. Dr. Shamoo's research lab studies the dangerous rise of multi-drug resistant bacteria. So we are very excited to have him today, and Dr. Shamoo, we hand it off to you.
All right. Well, good afternoon or good morning, depending on where you are, I suppose. Let me tell you about this project. It's a really fascinating project, and it started, this is actually a Defense Threat Reduction Agency project that I'm reporting on today, and it's a collaboration in part with Sandy Gibbons at ECBC, which I think has now been named the Chemical Biological Center now, so it's had a name change. But I think what I'm going to tell you today are 3 different stories that have to do with how multi-drug resistant pathogens come to rise in clinical settings, and in particular with an emphasis on warfighter situations. So as you know, warfighters encounter a lot of novel environments, and so one of the things we were interested in studying in this particular project is the rise of an emerging organism as it transitions from being something you've probably not heard much about, in this case the organism Nocardia nova, as it moves into the hospital or clinical setting. So that's our first story. Second one is actually a story around how hypermutation, the ability of cells to become very rapid mutators, is actually another way they drive themselves to becoming multi-drug resistant. And that's a study we did in Pseudomonas, which is a common pathogen in cystic fibrosis patients, for instance. And the third story really touches on some of the challenges we have around potential WMD agents, in this case, Francisella tularensis. And we were interested in understanding whether combinatorial therapies towards Francisella might be more useful than the single therapies, and what the hazards might be of using combination therapies for that particular organism. So it's 3 different vignettes, and I'll try to go through them really pretty quickly, but hopefully there'll be some good questions at the end. All right. So I need to tell you a little bit about how we do experimental evolution, because the key to this project is moving from a reactive mode of thinking about drug resistance to being one in which we can accurately predict how drug resistance is going to occur, and how we can move from sort of reacting to bugs as they show up in the clinic to one in which we can actually understand and predict how they'll become resistant. And then if we understand that, we can work towards preemption, the design of drugs that would prevent the rise of drug resistance, or would be more effective in combination with current antibiotics. So the idea is to move from reactive status to preemptive status. So the way we do our experimental evolution experiments, these are all done in vitro. So we have a series of rather sophisticated bioreactors that we inoculate with a starting population of susceptible bacteria. So if it's Pseudomonas, the Pseudomonas goes in the bioreactor. If it's depending on the organism, and they start as sensitive to the particular drug of interest. And then very slowly over time, over the course of usually 2 to 3 weeks, we slowly increase the drug concentration in the bioreactor, and then the organisms adapt. And so if you sort of look at this top panel here, you can imagine you start with an ancestor strain, which is very susceptible, and then slowly mutations accumulate in their genome as they become increasingly resistant to the particular antibiotic. By the end of the experiment, you've got usually a population of bacteria which are highly resistant to the drug of interest. And what's interesting about these bioreactors is there's just not one answer to becoming resistant. There's usually several evolutionary trajectories, that is, several mutational pathways that can lead you to being resistant. So the bioreactor is not clonal. It's actually very polymorphic. It has a very diverse population of different types of genotypes in the reactor as they evolve. So you don't get just one answer, you get a lot of different answers, which is what we want. We want to see all the ways in which the bugs could become resistant to the antibiotic, not just maybe one way. And so the way the experiment works, and I think is very useful, is that each day of the experiment, so we start with 0 drug concentration, then we go up each day. Each day we take a sample of the entire population, and we do deep sequencing on that population, so that we get an idea of what mutations are happening in the population on day 1, then day 2, then day 3. And from that, we can sort of, if you look at that top circle there, you can see you can build the evolutionary trajectories of how they become resistant. So you get the order of mutations that happen, so the first gene A, then gene B, and then gene C, as well as how common those particular mutations were in the population. So how good was that answer to becoming resistant? And that's very useful for the analysis if you want to identify what it would be a good drug target, for instance. And so the first question that people ask me typically is, well, does this match what happens in clinical settings? And the answer is yes, it really does. You do get occasional red herrings, but it's pretty rare, to be honest with you. We've published many papers on many different organisms and many different drugs, and they match very well with the clinical findings in patient populations. I would say what I would be careful about is we don't study pathogenesis. So pathogenesis, the establishment of an infection, is a different process. And so if you wanted to study pathogenesis, you would have to use an appropriate animal model. We're actually studying the biochemical mechanisms of becoming resistant. So it's a little bit of a distinction there, but it's very important, especially to my colleagues that are working in pathogenesis. So my background is in biophysics. So at the end of the day, what I really like to do is identify the mutations and then map them onto a 3-dimensional structure and understand how the mutation in the genome gave rise to a difference in the function of the protein that gave essentially the drug resistance, allowed the drug resistance to happen. So the end product of all of our work, if we're lucky, is a very, very deep understanding of how the mutations have arisen in the population, and then exactly what they did in terms of the biology, the in vivo biology of the organism, and its atomic root. So they're shown here are some structures of some molecules that we did where we've sort of gone at that problem. So that's where I like to end up, but that's at the end of the road. So the way the pipeline works in my laboratory, sort of in these boxes here, is that we use experimental evolution to generate populations of cells with drug resistance. we deep sequence those populations each day to identify which mutations occurred first, second, and third, for instance. And then we isolate those proteins if they're good targets, and we think about how we would actually design drugs against them. So if you know that, for instance, all of the particular cells become resistant as a first step, say gene A looks like a great target for drug design. But if that's one of 5 different opening moves, then maybe that's not going to be a great target. So our goal is to actually develop drugs that would actually work in combination with classic antibiotics, and therefore they would sort of work against evolution. So they would prohibit the organism from becoming resistant to that particular drug, so it maintain the efficacy of the antibiotic for longer. So that's the idea of where we're trying to get to with all this. And of course, as you identify the mutations in these populations, they also become clinically useful in terms of being molecular diagnostic tools for clinicians to say, is my particular organism resistant to this particular antibiotic? So there's a very practical side to all of this as well. So let me tell you the first story. So the first story is an organism called Nocardia. Unless you work in cancer biology, you probably haven't heard much about it. There's a little bit of a controversy in the field as to whether it's an emerging organism or just was poorly recognized in the past because we didn't have really good tools for doing molecular diagnostics, and the organism grows really slowly in the laboratory. So it's a very common organism. It's a soil bacterium found ubiquitously in soil and water. So if you're a gardener, you've probably been exposed to it many times, but if you have a good immune system, it hasn't really had any effect on you. So it's an opportunistic pathogen. It's typically seen in immunocompromised individuals, cancer patients, for instance. But interestingly, about 1/3 of the patients that arrive with nocardiosis or infection of nocardia do appear to be immunocompetent. So there's something we don't really understand about the particular organism. The treatment of choice for this particular organism is a combination of drugs, trimethoprim and sulfamethoxazole, TMP-SMX. It's a very old drug combination and is usually quite effective. We were interested in understanding how the organism becomes resistant to this particular drug combination. Now, when you're working with a new organism for the first time, and there wasn't that much known about nocardia, there's some very nice early papers out, but not much in terms of the genomics. The first thing we had to do is assemble a genome using long and short reads for a reference organism. And then because we didn't have a very good understanding of the diversity of the genomes of these organisms, we actually did evolution experiments on 10 different strains of nocardia, 7 of which were isolated from patients at MD Anderson Cancer Center over here in Houston. So we really wanted to explore as wide a range of mutational strategies as possible in this genus of organisms. So we did the experiment in 2 different ways. We did it through our bioreactors, which I told you about before, and we also did what are called serial transfers. That is, we grow them in flasks each day and increase the drug concentration in steps along the way. That can be useful because you get a lot of replication that way because it's easy to do 8-10 populations at a time as opposed to running. I mean, all my students would quit if I made them run 8 bioreactors at a time. So this is how TMP-SMX works. This is your classic biochemistry pathway to making folate, which is required for bacteria. And so what's shown here is the pathway and the enzymes that would be used to make folate. And so the drug works, sulfamethoxazole inhibits an enzyme called DHPS, or dihydropteroate synthase, and TMP, or trimethoprim, inhibits the enzyme dihydrofolate reductase. So it kind of hits the pathway to making folate in 2 different positions. And what's shown in the green stars are just sort of a summation of all the genes in which we found mutations that could give resistance. And so maybe not surprisingly, I don't think it's surprising, one of the enzymes that you can get mutations in that confer resistance is in these 2 target enzymes. So the DHPS is one, and DHFR, dihydrofolate reductase, is the other. So that's makes a lot of sense, right? You get mutations in the genes that the drug binds to, the proteins that the drugs bind to, and therefore that can give you resistance. What was a little surprising was we saw mutations outside, peripherally, in this pathway. Among the most interesting and most common, actually, was a mutation in a homolog of that dihydroteroate synthase. So the gene is called FOLP that encodes that synthase. And so there's another copy in the genome called FOLP2, which is shown over on the right of this picture here. And interestingly, mutations in that homolog also produced resistance, even when you didn't have mutations in the FOLP gene. So that was very unusual and very unexpected. And we'll get to the bottom of a little bit of what we think is happening there as we go on. So we'll start with the easy things first. So the mutations in dihydrofolate reductase pretty much map to the active site of the enzyme. Now, we don't have the crystal structure of the Nocardia enzyme yet, but there's a good structure for E. coli and for Mycobacterium tuberculosis. And what you can see in this picture here, the drug is shown in orange, TMP, and the substrate is shown in yellow, and the mutations map pretty much right at the active site of the enzyme. So what's been seen in tuberculosis is these mutations typically allow the enzyme to bind the substrate with a higher affinity than the drug. So it sort of reverses the interaction affinity of the molecule for the active site. So and that makes a lot of sense. The mutations are clustered right at this active site to confer resistance, so the drug doesn't bind as tightly anymore. So that makes some sense. And then in the case of the other enzyme, the target for sulfamethoxazole, it's a fairly similar story in that DHPS enzyme, where the mutations tend to map. In this case, the drug is shown in yellow, and the substrate, paraaminobenzoic acid, is shown in purple. Mutations, again, tend to cluster in the active site. So that phenylalanine 28 there is is very proximal to where the sulfamethoxazole binds. Some of the other mutations are a little further away, and those are kind of interesting because they must reconfigure the dynamics of the active site in an interesting way to disfavor the binding of the drug. And like I said, we're pursuing the crystal structures of these Nocardia proteins as we speak, actually. So those kind of make sense. You make mutations in the genes, in the proteins of the target of the drugs. What's less clear, though, is how you get mutations in a gene which is actually not in that pathway. So I mentioned to you that there's a second copy of there's a homolog of a dihydroterate synthase called FOLP2. It's about 36% identical to the first copy, the functional copy. And people did some really nice studies in tuberculosis some years ago showing that the second copy in the genome does not carry out the reaction, is not part of the folate pathway, and they actually couldn't even find conditions where that gene was expressed. So they actually said that, well, they made the protein and they showed that it could not carry out the reaction. So this homolog, whatever it's doing in the genome of tuberculosis, is clearly not doing anything in folate synthesis. And so what they predicted was if you're doing drug design, you don't have to worry too much about that homolog because it doesn't do the reaction. And in our case of our organism, in Nocardia, we see clearly that mutations in that second copy can actually do confer resistance. And so we're trying to figure out whether that means that the enzyme has now acquired the ability to do the reaction, or whether it's acquired the ability to inactivate the drug in some way. So we don't know that yet because we're still doing the kinetics and the biochemistry, but I think it has to be something like that, we think, where somehow this other copy is acquiring the ability to do the reaction or acquiring the ability to inactivate the drug, for instance. And that's, I think, really interesting because that was not predicted from the tuberculosis study. So it's always a cautionary note when you drawing conclusions from something saying an enzyme can't do something. Well, mutations happen, evolution happens. They can start doing different things. The other thing that was kind of interesting in the case of this second homolog that I was telling you about that was thought to be not important to the folate pathway is that one of the most common ways that the organisms became resistant was actually by an insertion of a C in the gene that encodes this enzyme FOLP2, this sort of, in theory, non-functional folate enzyme. And so it's not unusual in bacteria to have overlapping genes. And what this mutation did is it actually fused the first gene, FOLP2, to the second gene. It makes a single long open reading frame that makes this very long protein that somehow, when you make this very long protein, again confers resistance. And we have, to be quite honest, no idea how that's working at this stage. I mean, what's clear is that we've made, through this mutation, a new bifunctional enzyme. And the question is, how and why does that work? And I just have here, I know it's very wordy, but I think the point is that second gene, GPGS, that it now becomes fused to, it has nothing to do with folate synthesis. In fact, its activity in general in vivo is underdescribed, I would say. It makes methylated polysaccharides. It had nothing to do with folate. So we have no idea why making this into a bifunctional enzyme actually is conferring resistance. So that's, again, part of what we're studying on the protein chemistry side of things. So evolution gives you surprises, and I don't think any of us are surprised about that. The other thing that was really different about this organism is what's shown on the left. Now, let me just sort of walk you through that slide on the left side. This is an experiment we did with Acinetobacter some years ago adapting to tigecycline. So the bottom, the x-axis, are the days in the bioreactor that we're doing the experiments, so from 0 to 26 days. And then at the top, in the sort of bluish colors, the sort of bluish patches in the top, is our mutations and their frequency in a particular gene called ADES, which is known to be part of the tigecycline resistance pathway for Acinetobacter. And so when the blue guys, they're all different point mutations in the ADES gene. So the light blue is at one SNP, the dark blue is a different SNP, and they compete against each other. And it just shows you that there are multiple mutations can happen in this ADES gene. But the point of the graph is to show you can see how the area of the blue color, for instance, is how common that particular mutation was. And so if you look on the left side, the blue mutations occur first, and then the greens, and then the reds, and then the sort of turquoise ones at the bottom. And so bioreactor experiments allow us to explore the first things that happen are in this ADS gene and this GNA gene. And it's very, very beautiful in the sense you can get this very nice march to resistance, and you can map what mutations came first, second, third, etc. We didn't see that at all for Nocardia. Nocardia was an absolute mess. We saw mutations coming up. We saw mutations coming down. It was really, I would show you the data, but it would be worthless to you. So we were stuck with thinking about, well, what's different? What's happening with Nocardia that's so different than some of the other organisms we've worked on? And I can sort of visually represent something unusual about Nocardia in this picture here. So on the left is the ancestor strain. So Nocardia is a beautiful organism, forms very nice, very white colonies, as shown on the left side. And on the right is the result of our experimental evolution studies, either in the flask, shown in these two lower right panels, or in the bioreactor in the upper right panel. And when my postdoc first showed me this plate of the adapted population of Serogeorgica in this upper right corner, I said, well, you've got contamination all over the place. They look at this. There's huge colonies. There's gray colonies. There's, coastal looking ones. It's a mess. That's the worst looking cross-contamination I've ever seen in my life. And then she picked colonies, sequenced them, and they're all Nocardia. Every damn one of them. So this is a highly polymorphic population. So what's happening in the bioreactor is a very diverse set of phenotypes, which are not what you see when you do sort of classic flask transfer experiments. So that was a little bit of a hint of what was going on. If you actually look in the bioreactor, and I show these pictures just to sort of make the impact here. So on the bottom panel here, this is a picture of the bioreactor growing Nocardia in the presence of drug on days 6 through 23. And what you can see, it should be a nice clear glass vessel. And we do see biofilms all the time in our other experiments with Acinetobacter and other organisms, but we don't see biofilms like this. If you look in the day 9 picture or the day 15 picture, you can see these white, they look like stalactites hanging from the top of the bioreactor vessel. These are actually Nocardia biofilms. They're waxy, they're thick, they're very hard to break, they're almost brittle in their waxiness. And so this organism forms really, really pernicious biofilms. And I think that's part of the reason why it's very hard to clear from the lungs. In part, they form these really strong biofilms. And it's also one of the reasons why the population is so diverse, because there's a lot of different genotypes that are existing, in subpopulations within the bioreactor. So the biofilms are acting to shelter different genotypes and allow this rise of a very, very diverse community that you don't see typically. And so our conclusion from that study was that you have to be very mindful of the formation of biofilms, because they're obviously, I think many of us know, very important to becoming multi-drug resistant. In the case of Nocardia, they form these very waxy biofilms, which are very hard to probably make it harder to treat with the antibiotic, and also facilitate the growth of very diverse communities of bacteria, all being Nocardia in the particular vessel. So that was an interesting study because Nocardia was it adapted reasonably well to the bioreactor, but it was still a slow grower even at the end of the experiment. So that was our take-home message there, and also about that accessory homolog that actually ended up being a pathway to resistance that I think nobody anticipated. In our second study, which is a little bit, I think most people know a little bit more about Pseudomonas. It's responsible for long-term infections. We were interested in studying how hypermutation drives adaptation to drugs in Pseudomonas. So hypermutation is just this phenomena where you get a mutation, usually in a DNA repair enzyme, that leads to the accumulation of more mutations in the population just because DNA replication is not as accurate, and therefore you accumulate mutations just willy-nilly everywhere in the genome. Now, of course, many of those mutations, the vast majority of the mutations you're generating are actually not helping you adapt to the drug, but are just random mutations. So you get a lot of just mutations everywhere in the genome. But it still allows you to roll the dice more in terms of finding that adaptive mutation that does confer resistance. So hypermutation is a great strategy for becoming rapidly resistant to a particular drug, for instance. However, it's a bad bargain for the organism because as you accumulate mutations, everywhere in your genome, of course, you tend to decrease your overall fitness because you're just making mutations everywhere. And so it's a bit of a devil's bargain. Yes, you're becoming resistant really rapidly, but once the drug is removed, you would expect that your fitness would be really bad because you accumulate all these other mutations. And one of the things that people observe a lot in the clinical community is that Pseudomonas infections recur with great frequency. So you can treat them for a while, you can sort of put them into remission for a while, and then they tend to sort of come back and reinfect the patient quite aggressively. So we were interested in that phenomenon, whether hypermutation could help explain the formation of subpopulations that could help invade the host, in this case the patient. So in this experiment, I'm just going to, again, just to orient you, what we've done on the x-axis here is we've linearized the circular genome of Pseudomonas, and on the y-axis it goes from day 1 at the top to day 26 at the bottom on the left panel. So that's one of these bioreactor runs. And then the little blue hash marks are where the mutations occur within that genome. So you can see, like at the very beginning of the experiment, like at day 1, there's a really strong, like little blue mountain ridge over there, which is a lot of mutations accumulating in the population, and that's actually in a phage that's incorporated in the genome of Pseudomonas. And so it mutates rapidly. The phage are expressed in small amounts, and it's quite diverse in terms of genetic sequences, as phage are wont to do. When we introduce the drug on day 2, actually it induces the phage, and so you get this huge phage bloom, and it nearly wipes out the population. Then the population slowly comes back over several days and reestablishes itself. But then on day 10, you get a mutation in one of these DNA repair enzymes. And once that happens, then you can start to see, as you go from day 10 all the way to the end, you just see mutations accumulating everywhere in the genome. And that's seen in the second run of the bioreactor. The formation of hypermutators in the Pseudomonas population is really common. It can even happen when you don't have the drug present. They just easily fall into this hypermutator lifestyle, which seems like a really bad decision, but of course bacteria don't think very much. They just act in response to selection. So one of the problems you have when you have a hypermutator is you have mutations kind of everywhere, and you need to figure out which mutations are actually adaptive and which mutations are just random. So you need to sort the wheat from the chaff, basically. So for instance, in 30 endpoint isolates, or 29 endpoint isolates of Pseudomonas in one of our experiments, they had accumulated 761 total mutations affecting 563 genes. So you're not going to be able to do genetics to sort of test all the combinations of those things to find out which genes are actually the adaptive ones and which ones are just junk. So you have to take more statistical approaches to understand how to sort the wheat from the chaff. And in the course of the bioreactor experiments, where you're sampling the whole population, not just endpoint isolates that are single genomes, we had almost 1,200 genes mutated in those populations. So it's really difficult to figure out which ones are adaptive. So the way you do it is you usually take a statistical approach where you say, well, what is the probability that a random mutation will happen in a gene of a known sequence? And assuming that mutations occur reasonably randomly in the genome, which is an assumption, which is not particularly true in some cases. If you see mutations accumulating in a gene much more than you would expect, and I've circled here PMRB because this is a really well-known gene that's responsible for acquired resistance in Pseudomonas, what you can see is you get many more mutations hitting PMRB than you would expect by chance. And that identifies it as probably an adaptive mutation. Okay, so that's sort of how you have to sort through this very complex data. And then on the case of PMRB, this is kind of an interesting evolutionary story. This is the sort of, it's a sensor kinase, and it upregulates genes that confer the resistance. And on the bottom, in panel B here, what we're showing is a linear representation are all the mutations that have occurred in PMRB. So we've got our data there, clinical data, other people's experimental evolution studies. And what you can see, there are dozens and dozens of mutations that occur that can confer resistance in this single gene. So there's not one mutation, there's not, there's dozens of them. And that's sort of a nightmare from a diagnostic point of view, from molecular diagnostics, because any one of these mutations could be the one that gives you resistance. It also indicates, though, from a sort of evolutionary biology perspective, that you don't need a very strong change in function of this sensor kinase to produce the resistance. And what do I mean by that? If you need an enzyme like a beta-lactamase to become 100-fold better at doing its reaction, there's only going to be a very small number of mutations, if any, that could change your KCAT, say, 100-fold. On the other hand, if I need to make a 1.5-fold change, well, there's going to be lots of ways, lots of mutations that could do that. And so when you see lots of mutations accumulating in a gene like this, what that suggests is that actually it's a small gain or loss of function, which is driving the resistance in that case. So this is a nice example of, in this case, dozens of mutations, and it's not by no means complete of all the mutations that show that this is probably requires a very small or modest change in function of that particular sensor kinase. Now, as I mentioned to you before, when you become a hypermutator, it's a great way to become resistant. And what this panel shows you is actually, it's a lot of data, I know, but let's just focus on the gray line is the ancestor, the sensitive strain, so it doesn't grow in the presence of the drug. That's panel A. And the colored lines are the adapted strains. So the adapted strains, the benefit that they have in life is that if the drug is present, they grow reasonably well, and the organism lives to fight another day, basically. If you remove the drug, and that's shown over here, if you look at where the drug concentration is very low, then these organisms don't grow very rapidly. So, like I said, they've accumulated mutations in their genomes that have actually made them very resistant to in this case, the drug, but have become extremely poor in terms of competing against the ancestor. So they actually don't grow well. If you take away the drug, they're kind of losers. And this brings up another interesting phenomena, which ties back to our nocardia story. Pseudomonas, they form very nice biofilms, and what's shown in this panel A here, they make a very nice green pigment, and you can sort of see on these plates, depending on the endpoint isolate you pick, they're quite diverse. What's shown in panel B is the extent to which the different isolates can be drug resistant. So the lighter colors, low drug resistance. In the case on the left side, 12 strains or 12 endpoint isolates had almost no resistance, and some had very strong resistance, shown in the sort of dark color. And so what this shows you actually is kind of a surprising result. You grow the bioreactor at very high drug concentration for nearly 5 days. And at the end of that experiment, when you pull out colonies and you put them on a plate, about 10% of them or so are still sensitive to the drug. So they are actually a subpopulation, presumably being protected by the biofilm. They're not persisters because there's too many of them. There's about 10% of the population. They're sort of hunkered down in the biofilm, waiting for the drug to go away, and then they can act to reinfect. So you have this reservoir, this pool of Pseudomonas just waiting to reinfect. And the thing that's bad about them is they're actually not the ones that have hypermutated. They're the ones that are the ancestors that they have very fit genomes. So when the drug is withdrawn and all the sort of darker colored ones kind of don't compete so well, these guys can reemerge again and reinfect and allow you to restart the whole resistance game over again. So we think that's particularly relevant clinically. Now, the last story is one in which I think is particularly relevant to the defense mission, which is thinking a little bit about Francisella. And so I think in this particular audience, a lot of you know about Francisella, and you know a fair bit about it. It's a select agent. It's a potential bioterrorism agent. And we don't have a BSL-3 laboratory, so we don't work with the actual tularensis strain. We work with the live vaccine strain, Holarctica. And we were interested in understanding whether we could improve on the current practices in terms of limiting the formation of resistance in this particular organism. So the typical treatment strategies that have been used, doxycycline is very common, approved for FDA for treating tularemia. CDC stockpiles doxycycline and ciprofloxacin or Cipro for mass casualty settings. And so, both Cipro and Doxy are used for this organism. And we were interested in knowing whether you should use Doxy first or Cipro first, or whether you could use them in combinations to get more effective clearance of the organism, or potentially to limit the rise of multi-drug resistant strains. So the first thing we did was the sort of obvious control experiment. This has been done by others years ago, but you can just study the adaptation of the live vaccine strain to Cipro. That's shown on the left panel here. And the red line is the clinical breakpoint, so that's used clinically. So if you look at the graph, around day 27 or so, the populations of the bacteria are above the breakpoint. So it takes about 25 days to become resistant to Cipro. That's pretty typical, to be honest with you. It's an unfortunate truth that in my laboratory, we've now studied many drugs, many organisms, and evolution always wins. And it usually takes about 2 to 3 weeks for something to become resistant to a drug, with very few exceptions. On the right is the adaptation to doxycycline. And again, it takes it actually around, this is a good example of a very effective drug. It takes it over 40 days to become resistant to doxycycline. So that's good performance, to be quite honest. And so we were curious to say, well, if you become resistant to doxycycline, does that make it easier or harder for you to become resistant to ciprofloxacin, for instance? I'll get back to the questions at the end, by the way. And I'll go back for a second here. So we're gonna look first at where the mutations arise in these simple single drug experiments. So in the case of ciprofloxacin resistant strains, it's well known that Cipro hits gyrases, topoisomerases quite commonly. It's been well studied in E. coli and a host of model organisms, including, it's showing on the right here, a structure of Staph aureus gyrase complex with Cipro and DNA. Our mutations are kind of what you would expect. They're in these genes. And so in that sense, the mutations, if you can sort of squint your eyes here, you can sort of see these mutations accumulate near the DNA binding site and near where the Cipro binds, and that's sort of expected from other organisms like Staph aureus and E. coli. So that result it's not surprising. We're still characterizing these mutations as best we can. In response to doxycycline, doxycycline works by binding to the ribosome and reducing translation rates. So it binds the ribosome, and the mutations here that cause resistance to doxycycline tend to be in the 30S subunit in the ribosomal protein S10. And shown on the right here is a very simplified picture. This is supposed to be a ribosome, but if I showed you the whole ribosome, you wouldn't be able to see anything. So we've stripped away all of the other proteins, ribosomal RNA, except for S10, the 16S part of the 16S ribosomal RNA shown here in the gray, and where the drug would be presumed to bind. This is tetracycline. In this particular complex is where we think doxycycline binds as well. And the mutations that accumulate that confer resistance are actually in this very little loop here, shown by the purple colored side chain here. And this loop is actually, we've seen this mutations in this loop before. We had seen it in Acinetobacter baumannii adapting to tigecycline in a previous study. So these mutations are interesting because they're actually on the other side of the ribosomal RNA helix. The drug sort of, you can't get good depth perception there, but it's on the other side. And so what we think is happening is it's perturbing the RNA structure just slightly to kick out or to reduce the affinity of doxycycline for the ribosome, conferring resistance. So I see the questions here. I'm going to get back to those. Those are good questions, though. So in this experiment, what we did is we said, well, if you become resistant to doxycycline first, and then you put Cipro, do you see any big changes in how rapidly they adapt? And so if you become resistant, say, to doxycycline, for the panel on the upper right, what you find is it takes them a little bit longer to become resistant to ciprofloxacin, although that's probably because they're actually less fit. They grow a little bit more slowly, so I'm not sure this represents much more than a sort of change in the fitness of the organism. But the overall effect is it makes it a little bit harder for them to become resistant, but not appreciably. They still rise pretty rapidly in general. And then we tried the idea of trying to use both doxycycline and Cipro together to see if that would really delay the onset of resistance. And what we found, this is shown in the bottom panel here, is that instead of taking about 30 days to 40 days to become resistant, they took about 50 days. And I would say that's not particularly good as a combination because the problem with the combinatorial therapy is you've used your two bullets, right? You've used your Cipro and you've used your Doxy. They've become resistant, and now you're really in trouble. So we were kind of surprised that there wasn't more synergy between doxycycline and Cipro because they work by very different mechanisms. So the Cipro hits the gyrases and topoisomerases, and doxycycline hits the ribosomes. So we sort of thought that using them together would be much more effective than using them separately. But I would say at this point, the evidence would suggest to use the drugs sequentially. But again, these are not, I wouldn't change medical practices based on an in vitro experiment. Let's put it that way. And so, what I want to leave you with is this idea that we can predict emergence of resistance very reliably. So I think the take-home messages are that, these in vitro techniques, especially when they're combined with animal models and animal studies, we can detect resistance and the ways in which organisms can become resistant quite reliably and reproducibly. And that, of course, the strategies towards resistance are varied with the pathogen and the drug. That comes as no surprise. What we imagine is a world in which instead of using broad spectrum antibiotics, we could use more targeted drugs and kill the organisms we want to kill and not wipe out a person's entire microbiome, for instance. So the idea is, and this is some work we did with enterococci, is that you identify a particular target protein, you can develop a co-drug to inhibit the move towards resistance, and then you can use that to induce hypersusceptibility. So in the case of enterococci, we showed that by knocking out a particular protein called LIA-R, which is a part of a signaling cascade, the organism becomes hypersusceptible to the drug of choice. In this case, it was daptomycin, and that they really have a tough time becoming resistant. It's very, very hard for them to become resistant. So you would use these co-drugs with an antibiotic to maintain the efficacy of the antibiotic in the patient. And so we sort of imagine this would be kind of a more like acting, with precision, right? So if we have precise diagnostics to tell us what the organism is, then we can actually target drugs towards those organisms instead of sort of taking a sort of carpet bombing approach of the broad spectrum antibiotics used, which is just to wipe everything out of. But of course, doctors need to know what, we need to have rapid diagnostics for this dream to come true because, when the patient comes into the hospital and they've got a bacteremia of some sort, broad spectrum antibiotics tend to be the things you use because you don't know what you're dealing with just initially. So I understand clearly that we need to get better diagnostics. But I think in the future we will have those rapid diagnostics, and if we pair that with really selective drug agents that hit particular organisms in their weakest spot, in their Achilles heel, then we can actually clear that infection much more rapidly. So with that, I need to thank the people who actually did all of the work because the lab, of course, they never let me near the bench anymore. And this work, as I've mentioned, was sponsored by the Defense Threat Reduction Agency. It was a collaboration with Sandy Gibbons. ECBC, now renamed, and of course we also support from the NIH for our work on enterococci. So with that, let me look at the questions a little bit more carefully here. The first question is, do you start with a polymorphic population or just end up with one? That's a really good question because when I was a grad student, I was taught you have a colony on a plate and it's a clonal. I think basically once you grow that cell up into a bioreactor of a couple hundred mils, you've got some pretty good standing diversity. Clearly, the stress of the antibiotics on that population increased the polymorphism dramatically. But even if you pull out, sequences initially, you'll see some polymorphism, but you get a lot more in response to the antibiotic, for instance. So you end up with one that's very diverse. In the bioreactors, in the flask transfers, they tend to be less diverse. And I think that's because in the flask transfers, you don't get complex biofilms. A flask transfer experiment works by you transfer cells each day to a new flask. And that means any cells that actually stuck to the glass and formed biofilms are disadvantaged. They don't go into the next day. So actually you work against biofilm formation in a flask transfer experiment. But in a bioreactor where they're all the same bioreactor for weeks at a time, then you can get really good biofilm formation, long-term biofilm formation. And I think that gives you a much more polymorphic population. Do we look at KCAT? We look at everything we can measure. So for these enzymes, I will tell you for the nocardia enzymes, FOLP, the dihydroterate synthase, we've been having a heck of a time just expressing that protein. So we've spent the last 6 months trying to make that protein for crystal structure studies in biochemistry. So we will measure the KCATs and the KMs when we get them. In E. coli, There's some very nice biochemistry done on the adaptive mutations. So I expect it's going to be the same or similar in many respects. I don't recall off the top of my head whether they're more driven by K-cats or K-amps. Now let's see. Do we have viable but non-cultural bacteria in a biofilm? That's a tough one. That's a tree falling in the forest. Do I hear it or not? We should be starting with, the things we start with are a single organism, Nocardia serrageorgica. So they were culturable at the beginning. We imagine they're culturable at the end. I could not prove that to you, though. I don't know. Yes, and so the other question is, do some antibiotics potentiate? Yes, of course, yeah, as you say here, some antibiotics do potentiate the activity of others. And we see a lot of that actually not in these studies, in our work with enterococci and daptomycin, which is a cyclic lipopeptide that affects the cell membrane. There are definite connections between the synthesis of the cell wall and the formation of the membrane, the biosynthesis of the membrane. So these things are often very tied together, as you point out here. So I think the answer is yes. It sort of depends on the specific antibiotic. Yeah, of course, we can provide the references. I mean, you can just shoot me an email if you like. I'm Shamoo, Shamoo, @rice.edu, and I'd be happy to give you the references for this work. Because yeah, it's true. I just gave you the one-liners. And then Sanjay asks the DHFR. Yeah, so we haven't done that. We have not mapped our DHFR mutations onto the other known ones, except the ones at the active site. I actually did a lot of work on adenylate kinase as an assistant professor, and we often found mutations that were so far away from the active site that would confer changes in the active site. And, I taught biochemistry for a long time, and, I've sort of moved away from, I mean, it's nice when the mutations show up in the active site because it makes it a simple story to tell. But distal mutations can affect, of course, molecular dynamics in profound ways. And so, a lot of enzymes have, lid opening and lid closing. They have dynamics, of course. And so, you can make mutations away from the active site that I think you well know and appreciate that can change how they interact with the substrate or product release and things like that. So those tend to be a little bit harder to figure out. But we're always excited by them because I think in some ways they're the more interesting mutation. I hate to say it that way, but they're just more fascinating because they're not obvious how they work. Let's see. For the question about PMRB, are those mutations clustered in flexible regions of the protein? I think it's hard to say with any consistency that's right. I'd have to think about that question more carefully to see whether it's statistically true that they are overrepresented in flexible regions. There are many inflexible regions and some that are not. I don't know the answer. I'd have to think about it a little bit more. My first impression is I don't think so, but I don't know. I'm probably I need to actually do the science and think about it a little bit. And Sanjay asks again about the statistical method. Okay. That's a really good question. Okay, so so you've actually analyzed one of the things I sort of glossed over very, very quickly, which is in my statistical analysis said, well, if you get more than you expect, more mutations than you would expect in a particular gene, that's a good sign, a signature of adaptation. But what if there's only one mutation, right, that you just that just is the only way to say make that fivefold change in activity? So it's not overrepresented; it's one mutation, right? It's just there by having replicate populations. If you do 10 populations and you see that single mutation like G238S and beta lactamases, for instance, you see it over and over again and repeated in different populations, then you can assert more readily that that's probably going to be an adaptive mutation. But you're all right. You have to use a couple different statistical heuristics to get the best answer. The other thing that's a caveat to what I said, which you probably will pick up on next, is that one of the things that's most commonly mutated in those populations are actually a mutation to mut S, which is a DNA repair enzyme, which is what causes the hypermutation. But there is no way MutS has anything to do with drug resistance. It's just the first mutation that generates the hypermutation phenotype that gets you to be adapting very, very, very rapidly. But if it shows up really clearly in our analysis, but we have to sort of say, well, we don't think it's involved in drug resistance. We just think it's involved in becoming a hypermutator. So there's always little caveats there. That's a really good question. Let's see. Oh yeah, no, yeah. So DHFR, yes. So DHFR has a distinguished record, I would say, in the biophysical community as being a model enzyme for model bizarre behaviors. And so I won't pretend to be a DHFR expert. I'm going to have to, once we have the proteins made and the mutants made, and we've been prioritizing studying the hydrotherapeutic synthase homolog more because we think it was unusual that it was a bypass to becoming resistant. So I'm just fascinated by the idea of whether this enzyme that previously couldn't do the reaction of making in the folate pathway can either do the reaction or now has gained the ability to actually inactivate the drug. I just find that to be a fascinating question, change its structure and function. So we've been sort of pushing on the other enzyme, but I think we'll have to go into DHFR, but we'll have to go at it carefully because of its colorful history in terms of as a model enzyme. So I think those are all good questions. Let's see here. Okay. Oh yeah. So this Sante asked about epistasis, right? So this is of course very important to any evolutionary trajectory. Is whether mutations either in the same gene or across genes. To what extent do they work together to facilitate each other? And so we find that just speaking broadly, in these evolutionary trajectories, we have many cases where the opening move is conserved. So you see a mutation in a specific pathway as the opening move. They might make a different mutation, but they're all, say, turning up a signaling pathway in some way, for instance. We see that a lot. And then the second mutation is sometimes a compensatory mutation. So they've kind of made themselves less fit by that first step, and then they sort of correct a little bit in the second mutation. So sometimes you see not just epistasis between mutations that are better than the single mutations alone, but are actually second mutations, which when you sort of put them back in the organism do absolutely nothing for drug resistance, but only work in terms of increasing the fitness of the organism in the context of the first mutation. So typically we see, pretty conserved evolutionary trajectories at the beginning of our experiments. And then as you get towards the end, say day 20 or something like that, you see lots of different mutations. They're kind of small incremental changes. A lot of them are compensatory. Oh, I see what you're, okay. Whether we see, yeah. So that's an interesting question in itself because it talks about whether we could be forming what I would call the bona fide social interactions or communities within the bioreactor. And I think that would be extremely cool because those would be social interactions that we're evolving. And I think there probably are, because the problem is when you take something out of a biofilm and you grow it, it's absent its partners, right, in the complex biofilm environment. So there are reasons to believe, I think, and I think it has been shown in places like the gut that you can get, whether it's cross-feeding or cross-signaling, sharing of resources in different ways. I think we do have that in the bioreactor. We've been doing some really cool experiments using microfluidics recently, where we can put some of our isolates together in sort of emulsion droplets to sort of look at synergies or social interactions between the isolates. But I feel like as a betting man, I would say definitely we're getting the formation of what I call real communities in that bioreactor, and that's part of the story that's really hard to analyze because the way our bioreactors are set up, I can get at the biofilms at the end, but to do really proper biofilm studies, you need to sort of you need to grow them and stage them and sort of do cross sections through them because there's some beautiful studies of looking at how electron transfer happens through biofilms, how you deal with oxygen debt in biofilms. I think that's something I'd love to have a collaborator on because I'm not a biofilm expert per se, because it is a deep literature.
I think we will cap our questions there.
This
If
is
any
actually only.
participant has a, I'm sorry.
Okay, those are great questions.
Good, good. And if anyone has a
Those are great
follow-on
questions.
question, Please email us at info@hdIAC.org, and we will forward those on. Yousif, we want to thank you for an excellent presentation, and we want to thank our participants for very good and informative questions as well. We have a short survey on your screen, and we would very much appreciate your feedback on the webinar, and thank you everyone for attending.
Thank you very much for inviting me.